Well, somebody gave me some advice that, when you start this, what you should do is start from the end and then go back to the beginning. That way I kind of incentivize you to this is what we're going to do today. This is what we're going to discover today. At the end of today, what we're going to do is we are going to construct a meta-analysis. And what we are going to do is we are going to use the 748 transcripts that we had, but we're also drawing from 7 other different studies looking at the same activated versus germinal cell lymphoma. And at the end, what you're going to do is you're going to get a gene list like this. And if we go down here, what's the focus of the paper that we just analyzed? What were they looking at? The ChIP-seq, what was that? STAT3, right? If you go down here, here's STAT3, and if I click on this, just watch this, you will get this list. Here is what STAT3 did in our study. It went up 3-fold. But here are 7 different studies done by 7 different groups from around the world at different times. I now know what STAT3 is doing in all of these, the fold change and also the p-values, right? That would tell me STAT3 is definitely playing a role in this system. What we're going to do is we are going to get that. And what I'm going to basically explain to you now is that this list that I put together, all of these genes, if it's red, it means it went up in all of them significantly. If it's green, it went down. This list is better than the one we put in, right? And this is where we're trying to get to, is that why would I leverage just my data when I can leverage everything out there that's looking at the same dang thing, okay? So I'm going to tell you why this works, and it all has to do with crowd theory. There it is. Okay. First, a few disclaimers and disclosures. The opinions expressed in this seminar are mine and not those of Illumina. I'm sure I use their software in many different ways that they never thought to use it. Also, I do not get direct compensation from Illumina, but I do receive reimbursement for travel when I speak at their events.
And dinner.
What's that? And dinner, yes. It's always great to hang out with the corporate guys because they have the corporate cards, It's not like the NCBI people. So, yeah, every time they go out, I'm like, hey, where are you guys going? And then I just tag along. Okay. So, when we talk about wisdom of crowds, we're talking about this guy. He kind of popularized it back in the day. His name is Sir Francis Galton. Anybody hear of Sir Francis Galton? Usually I get one person. No? And then I go, hey, well, what did you do? And they're like, I don't know. Well, he was a cousin of Charles Darwin, and I think he was actually smarter than his cousin. He was greatly influenced by some of the work, obviously, Charles Darwin did in evolution. Just like all the cool scientists of the day, he was a tropical explorer, geographer, meteorologist, psychologist, and criminologist. And since Zoo went blue before me, I'm going to have to tell this story. So on one of these trips, he went to the Middle East. I read this biography on him, and they got some of the letters from these trips. And I guess he came back, and one of the guys that was hosting him said, hey, I'm sorry about, bad time on your trip. Next time we'll get you a virgin. So he probably got an STD on one of those trips. Actually, he would come back, and on these trips, he went to Africa, and, he basically figured out how to survive in the wild. This was basically, no one went to this, no white people went to this territory. And so he basically wrote this book on how to survive in the wild. And it was a bestseller in England, and he taught the English, he would teach the English troops, how to survive in the wild. It was pretty cool. But anyway. Well, the reason why you probably didn't hear of him, he's also known as the father of eugenics, right? And that's the belief that, there's some intrinsic, trait, or, people would measure people's heads, and, obviously people use race for this, that certain people are better than others, right? And what we're going to talk about, this wisdom of crowd, is that, it kind of disproves that the best isn't our outliers. It isn't, the outliers in humans. It's probably the average of everybody. And so we're going to talk about that. We're going to talk about finding the averages in different scientific studies. And that average is going to be better than each individual one of those studies. He was a geneticist. He pioneered the biometric approach. That's kind of the belief that if you measure a bunch of the crap out of something, take one object and measure a bunch of things about it, and take another object and measure the same bunch of things about it, and you compare those measurements, you're going to find a lot of differences. and it's going to explain the system. And I think he would be completely at home in today's genomic world, right? You know, we all have 3 billion bits of information associated with us. It's kind of the biometric approach. And the way he influenced me the most, he was as a statistician, he kind of expanded on the concepts of variance and standard deviation, which are huge in statistics. I think him and Fisher are probably the godfathers of modern statistics today. By the way, they both hated each other, which you could imagine. Also, he developed the concept of correlation, and we're going to use this today. How do we compare our study to everything that's also been out there, right? We're going to use this concept of correlation. And again, I'm going to get in more depth of the wisdom of crowds. He was a really smart man, and he influenced a lot of smart people. One of those was Carl Pearson. Has anybody heard of Carl Pearson? Pearson correlation test? Yeah, that's him. He actually wrote a 3-volume biography on his mentor after he died, which I have not read. I guess I like the smutty stuff or whatever. I don't know. They also influenced Charles Spearman. Spearman correlation test, right? Same kind of thing. It's the non-parametric version. And I found this out later. Anybody know who William Gossett is? He was a student of Carl Pearson. Anybody, the William Gossett, does that name ring a bell? What is the most famous statistical test? The most used, let's say that. Students' t-test, right? They don't call it a Gossett test, do they? No, I call it a student test. He was actually slumming. He was working for, he needed some money, so he was working for the Guinness Brewery. And he basically developed a way to use small sample sizes to determine, different aspects of the beer. And Guinness at the time wouldn't let him, they had a kind of a policy that you couldn't publish papers, because they were a company. So he basically begged them, and the only way they would let him publish the paper on the t-test was with a pseudonym. So he used student. So that's why we call it a student's t-test. You can say that at parties. I'm telling you, it's a good party, because you can get in the Guinness and you can bring in all this, beer and Irish. Okay, but the person you've probably heard of, has anybody heard of Nate Silver? Right? Yeah, 538. In 2012, he predicted almost every major election in this country. I think there was one election in North Dakota he didn't get, right? Do you think he asked anybody who they were voting for? No. All he did was take all the polls that were out there, right? Some polls obviously were biased left, some were biased right. He took the middle of all those polls and got the exact election results in 2012. Again, wisdom of crowds, right? We can do this with studies. There's good studies, there's bad studies. If I take the middle of it, it's better than all of them. And so we're going to do that today. Okay, so let's talk about the wisdom of crowd. And this happened when he was in his 90s. I love that. He was still thinking back, even at the end. So in his 90s, he went to this English fair. And what they were doing at this English fair was they were auctioning off the weight of a bull. There was this contest, the processed meat. And so they had a bull there, and what you had to do is guess the weight of when they basically butchered the ox, how much that butchered meat would weigh, right? So this was and people had to, pay money to guess. So they were motivated to guess correctly, right? No one's doing that's 3 tons, It was a 6 penny. I have no idea how much that cost. I'm sure it wasn't cheap back in the day. 13 tickets were rejected for being defective or ineligible. So, Galton saw this, and he thought this would be a great test of his eugenics, right? That he had a bunch of butchers and farmers that were also made guesses. And he goes, I know these guys are going to make much better guesses than the lay people that are also guessing. This was going to be his proof of the, kind of his eugenics. So he gathered everybody's guesses. He got all the, the papers that were submitted and kind of pulled them together. And what he found out is nobody got the exact weight of the bull, right? Some were really high, some were really low. But this was his aha moment, is when he took the average or found the median of all those guesses. It was within 1 pound of the weight of the bull, right? And so he was pretty, he was amazed. He was like, oh my gosh, I can't believe this worked. I thought my experts would be way more accurate. But by taking everybody's guess and finding the middle of that, it was much better than all my experts. And he said, maybe we should study this. And sure enough, for the last, and you can actually look up this paper. I put it in your, I don't know, what is it, the box or the, file sharing system. But, it's a page and a half. I love those days, back in those days. Like the sequence of DNA, that published paper from Watson and Crick was another, it was like a page and a half. So, but you can read it. It's a good read. So ever since then, we've kind of been replicating this whole wisdom of crowds, using this average. And there was this book that came out, The Wisdom of Crowds by James Sarawecki. Did anybody heard of that? It was a bestseller for a while. But again, he kind of goes into some of the ways we're using, wisdom of crowds, some of the, restrictions that you have to use it correctly. Again, I talked about Nate Silver. He has a website, 538. Go check that out. I love his website. It's a really data-driven news kind of website. But again, he's doing the same thing as, he's pulling from lots of different places and kind of finding the average of what everybody says. Even the CIA is using wisdom of crowds. That what they've done is they've gone average citizens and they'll ask them geopolitical questions like, what is the probability that Russia will invade Ukraine before November? And all these people, they don't give them anything else, and they just go on the internet, find their own information, and then they give their guess. When they take the average of all their lay people, all their guesses, it's much better than the experts that have been at the CIA for 30 years, right? This stuff works. Picture this, if we had a bullseye on this wall, and I had everybody in the hotel throw a dart at it, right? Maybe nobody hits the bullseye, but if I take the geometric mean of where everybody was throwing, the center of all those throws is probably the bullseye. That's what we can do with biology, and that's what I'm going to show you with this software. But there's there's requirements of this, is that you have to have diversity. You have to have lots of different people making guesses, right? We have that. This group that we're doing the analysis on is obviously not the only people that have looked at activated versus germinal B-cell lymphoma, right? There's other people out there looking at this. Also independence, that all of these people have to make their own uninfluenced decision. Now, as you know about labs, especially labs working in the same area, they fight with each other. They don't want the same opinion. I like that. That makes, using this kind of concept for research is great. And then specialization, right? Not all groups that are looking at B-cell lymphoma specialize in the same thing. You might have immunologists doing one type of study or they have their specific skill set. And you also might have some people that basically specialize in, cell cycle genes. And then aggregation. How do we take all these guesses and find the middle? And with science, with these studies that we look at, gene expression is perfect, right? Fold change, right? I can take the average fold change of all these different studies. We can condense, we can basically get all this information and boil it down to one number. Okay. So why the heck would I want to do this? Is because there's lots and lots of crowds in research. Does anybody seen this page, Gene Expression Omnibus? Anybody familiar with it? This is basically where we're dumping all our public data. Has anybody tried to get information studies out of Geo? Anybody? No? You're not missing nothing. It's hard. And it's terrible, right? If you look here, but there's a lot of gold in there. There's 74,000 different studies in there that we can possibly draw from, right? But again, it's hard to get to. What we would like is basically a convenient platform that has gone in there and grabbed this data, all the stuff we can use, and give us basically a platform where we can access it easily. And so that's what we're going to do today is we're going to use this software that has gone through Geo and basically pulled out all these different studies. So again, here's our experimental setup, activated versus germinal B-cell lymphoma. This is the paper that we're going off of. Again, this is a process. So anytime I I don't program at all. And I know most people, when they go, what do you do? I'm a bioinformatician. And they go, oh, what programming language do you use? I'm like, the button kind, right? I think sometimes, I think a lot of people get excluded from doing bioinformatics is because they think you need to code, and you don't, right? I think, for me, I would rather focus on what the tool could do for me than what the tool is, right? There's a lot of people that focus on the tool, but, I want to just take something and just run with it, right? And this software allows you to do it. So, again, I come from the bench, so whenever I get into a project, I basically do a little research, right? What is activated versus germinal B cell. What should I see? What has other people seen? I should see kind of the same things in my gene list, but this is a whole new, different analysis. I'm going to find different things as well. So what I use is I just kind of review what's going on. This is two different types of cancer from the same developmental B cell development. At different stages these cancers develop. These cancers have certain, molecular signatures, a lot of histone modifications associated with germinal B-cell lymphoma, EZH2 mutations, BL6 expression is to the germinal center, or the germinal cells. mTOR pathway should be activated in these, and then you see a loss of PTEN with the germinal cell. Here in the ABC, the activated B cell, we should see, these should be minus CD79. Again, histone modifications, BCL6 translocations. And again, what we've seen is that you have cytokine signaling, or JAK-STAT pathway activation. So what I should see is, in the activated form, I should see a lot of immunological signaling. Some of the markers used to characterize the two steps, or the two stages, CD10. So if I'm measuring CD10 in my activated phenotype, there's probably something going wrong. Again, this gives me an IRF is not in the germinal, but in the activated form, and again, BCL6. So, and then this is giving me some markers that I should see these differences between these two tissue types. But this is why we care, right? No one would care about these two different cancer types if there wasn't a difference in survival. And that's what it is. So if you had a choice of getting activated versus germinal B-cell lymphoma, which one would you pick? G, right? Germinal, right? Here's what we're doing today is what we're basically figuring out. We're basically determining what genes are different between those two types. And based on those differences, maybe we can determine why the survival is so different. Maybe we can do something to the ABC to make it more germinal-like, right? Again, all we're doing is analyzing our gene list. We're looking at a bunch of genes that are down in one group but up in the other, or vice versa, consistently up in one group and down in the other. And from these genes, we're going to basically pull out biological units or biological information that will inform us on the system. It's experimental design. I know I'm going over this, but again, we had N of 4 versus 4. We did some quality control. We took out one GBC sample. From that, we were able to put a list together using a false discovery rate of less than 5%, and we ended up with 748 transcripts. Okay? This is what we're going to start off. This is what we're going to analyze. Okay, we'll come back to that.